Hello everyone, welcome to the DMPK Unraveled Scientific Webinar Series, Episode 4. Today's topic is From Physiological Based Pharmacokinetic Modeling to First in Human Dose. How PBPK model is used to predict safe and effective first in human dose and drug-drug interactions. So before we start, some webinar etiquettes. Uh, Question and answer will be at the end. So if you have any question, please feel free to type in the chat box and we'll address your question at the end. So our first speaker, Dr. M. Surlevel Rajan from Manipal College of Pharmacy, he will speak on physiological based pharmacokinetic modeling and its application in drug development. Our second speaker, Dr. Claire Wilson from Syngene International, she will speak on human dose prediction, standard practices in the pharma industry. I'm Dr. Amol Raje. I'm working as a director in DMPK department of Syngene International Limited, and I will be moderating this webinar today. Syngene has a rich experience of 30 years in providing solutions to the global clients. We have around 400 plus active clients across the globe and around 8,500 employees work globally for Syngene. Syngene has $4 billion market capital, and Syngene till date produced 450 plus patents and created IP space for our client. Syngene is committed for its safety, data integrity, data security, and supply chain. Syngene team is expertise in various modalities like small molecule, peptides, oligonucleotide, antibodies and antibody drug conjugates, degrader, and CAR T cells. We are well experienced in various therapeutic areas like oncology, immuno-oncology, autoimmune disorder, neurosciences, metabolic disorders, inflammation, and niche indications. Coming back to objective of this webinar series, this webinar series has started to bridge the gap between academia and industry by sharing valuable insights and success stories in the field of drug metabolism and pharmacokinetics. This series aims to bring together thought leaders, academicians, industry scientists, and researchers across the globe to discuss the latest development in DMPK with a focus on overcoming key challenges such as druggability issues and optimizing molecules for next stage of drug development. To highlight real-world case studies and practical solutions, the series will foster collaboration and knowledge exchange, empowering participants to navigate the complexities of drug discovery and development more effectively. So what is physiological-based pharmacokinetic modeling? Physiological-based pharmacokinetic modeling is a mechanistic model where body compartments represent as body organs represent as a real compartment, like real organs like liver, kidney, brain. They act as a compartment, and this PBPK model integrates various physiological parameters such as organ blood flow with tissue volumes, and it integrates with various physicochemical properties of the molecule like lipophilicity, pKa, solubility to produce what will be the effect of various factors on the pharmacokinetic profile, physiological factors. It has wide applications across the drug discovery and development. So PBPK model is used for predicting first in human dose. It also predicts what could be the drug-drug interactions. It predicts the enzyme inhibitors or inducers effect. It is also used to predict a simulation based on co-administration. It also reduces the need for multiple clinical drug-drug interaction trials. PBPK model is widely used for predicting PK profile in special populations such as pediatrics, elderly patients, pregnant women, hepatic or renal impairment population. Formulation development scientists also widely use this application software for predicting the food effects. They compare what could be the PK profile of immediate release versus modified release formulation. they use this software for predicting the bioequivalence decision. PBPK model is also used to design the clinical trials. So what could be the effective dosing regimen, what population we need to select, what could be the population variability, and what exposure we'll get in various with different factors, that is also used for prediction. PBPK model is widely used for regulatory submission like dose selection, labeling for drug interactions, pediatric dosing recommendation, or sometimes it is also used to get the waivers for some clinical trials. You can see here, PBPK model is widely used for informing the dosing in drug labels for the FDA. You can see here the accumulative number of these submissions to the FDA has tremendously increased from 2013 to 2023 across various therapeutic areas like endocrine disorder, cardiovascular diseases, gastroenterology, infectious diseases, CNS, rare diseases, and oncology being the highest submission in oncology. So if you look at this graph, you can see over the past 15 years, PBPK modeling has become a key part of regulatory landscape due to scientific advances in this field and the increasing number of PBPK submissions from the pharmaceutical industry. Publications of PBPK-centric guidelines uh, by the FDA and EMA in 2018 set the stage for regulatory requirements and regulatory PBPK reporting. Due to this, currently PBPK approach is advocated in most of the FDA and EMA regulatory guidelines that cover the clinical pharmacology development of a new medicinal product. So with this short background about PBPK model, I would like to introduce our first speaker, Dr. M. Surlivel Rajan. Surlivel Rajan Maleswamy received his postgraduate degree from Tamil Nadu Dr. MGR Medical University, Chennai. He completed PhD in the area of population pharmacokinetics from Manipal University in the year 2011. He had his postdoctoral experience from University of North Texas Health Science Center, Fort Worth, Texas, USA. He started his teaching career in the year 2001 and has more than 25 years of teaching beside a brief stint in industry. His area of interest are clinical pharmacokinetics and population pharmacokinetic modeling and pharmacotherapy. He is the president for Society of Pharmacometrics and Health Analytics and a board member of Asian Pharmacometric Network. He has around 80 publications in peer-reviewed journals and 2 book chapters to his credit. He is currently guiding 7 PhD students and 6 PharmD students at his university. He is a reviewer for national and international journals. He received research funds to the tune of 50 million rupees from various government agencies and industry for carrying out his research as a principal investigator and co-principal investigator. So with this background, I invite Dr. Rajan to present his talk on physiological-based pharmacokinetic modeling and its application in drug development. Over to you, Dr. Rajan.
Thanks, Amol, for the introduction. Today, we will discuss about the PBPK modeling paradigm, which will lead us to the predictive pharmacokinetics. So, PBPK model is at the center of this mechanistic model concept we are going to discuss today. PBPK models are coming much earlier to the standard pharmacokinetic studies we do in the clinical trials or in the phase 1 studies. And this PBPK model can come even before carrying out animal studies. We call there are different approaches are there, bottom-up approach, top-down approach, and middle-out approach. I'm going to discuss about all those approaches. And using these approaches, we can predict the pharmacokinetics in animal models and use that information and scale it to humans as well. So when we conventionally look at the classical PK models, also known as empirical models, against the so-called mechanistic or PBPK model. The difference is that classical models just explains the pharmacokinetics, but there is no mechanistic explanation for that. There is no physiological meaning there in the system. But PBPK model explains why it happens, and the parameters are scaled using known human physiology and scaled in vitro data. This allows for direct extrapolation across species and diverse patient scenarios. The catalyst for industry adoption of this PBPK model is three-pronged. Advancements in vitro in vivo extrapolation techniques that allows for accurately scale the in vitro assay data into understanding the actual parameters, and computational tools, rise of powerful computational simulators, including SimCell, that usually helpful in the prediction of drug-drug interactions, and tools like GastroPlus and PKC, they will all handle complex multi-compartment model equations. Therefore, these computational tools have play a significant role in the development of PBPK modeling and their acceptance. Regulatory guidelines also are there available for FDA from FDA and EMA, especially on drug-drug interaction studies and reporting. With all these reasons, PBPK model is increasingly being used by different industries. So when we want to understand the fundamental concepts in the PBPK model, the mass balance differential equation, which tries to understand the rate of change of, imagine, drug concentration in any compartment within our organ system. The rate in minus rate out, it's again calculated using the blood flow inside the system and how much going out of the system, how much of drug goes inside the system, and how much of that goes out with the outward flow. And for eliminating organs, additionally, the intrinsic clearance is also counted. So that way, the physiological model, physiology-based model, can account for the drug movement in and out of any organ system with appropriate scales and volumes. So when it comes to the type of model, there are two different subtypes are there: perfusion rate limited and permeability rate limited. So for small molecules, the kind of model normally used is perfusion rate limited, because the small molecules can easily pass through various membranes and only the blood flow is the limiting factor there. Whereas in larger molecules and polar molecules, the processes like active transport, cellular uptake, efflux, all of that determines the drug movement. So that is predominantly controlled by the permeability rate limited. So these are two subtypes of the PBPK models available. When it comes to key building blocks of the PBPK model, organism properties, drug properties, and study protocols will influence the model. So when I say organism properties, anatomy and physiology, like organ volume, surface area, tissue composition, and so on. Drug properties, that will determine how the drug distributes inside the system, like lipophilicity, molecular weight, and so on. And study protocol, that depends upon the, again, the type of formulation and administration protocol, and special events like with food, without food. All of this will become part of the PBPK model inputs. So when it comes to biological variables in that system, specifically CYP450 enzyme abundance, transporter system, and the expression levels across the different tissues will also go into these inputs. Apart from that, variability, which is coming from different subjects, will be accounted by incorporating demographic and genetic variants. That means we can create virtual populations on how they respond variedly for the doses and drugs. That can be also be identified by using these Monte Carlo simulations. So when it comes to drug-specific parameters, the intrinsic clearance, the calculation of intrinsic clearance from in vitro assays, the data coming from in vitro assays, and that will be scaled to predict in vivo humans level clinical pharmacokinetic parameters. So that's how the drug-specific parameters will be scaled from in vitro assays to human level. When we are scaling, we take into account the considerations like microsomal protein per gram of liver and liver weight, and as well as the tissue distribution parameter like KP will consider the drug lipophilicity, ionization, and protein binding characteristics. This kind of predictions will dramatically reduce the need for costly in vivo studies in animal distribution scenarios. So currently PBPK modeling strategy involves kind of learn and confirm paradigm. I'll talk about that in detail. So from animal models, we can scale it up to human volunteers level, that is healthy adult human volunteers, human level model, and then scale it further into patients of different subtypes or special groups can be considered. That is how the PBPK modeling strategy is now involved. So the paradigm involved is learn, confirm, refine, and if needed, again relearn and continue to learn. By this way, what happened is that with the animal simulation data is taken to in vivo, and whatever predicted is coming very reasonably in the animal model, you can proceed further to human studies. Or if the prediction was not accurate, you go back and study the fundamental additional experiments carry out and improve the model further. This is how learn, confirm, refine model paradigm works. If the simulation at any stage is inaccurate, considering all the assumptions, this would indicate the violation of one or more of the model assumptions. That means we are going to repeat the experiments and understand the mechanistic pathway and redo the experiment till we get the predictions correct. So by doing this way, what actually we are doing is, actually prevent the late stage failures. So one clinical example I can discuss here. Repaglinide, a short-acting meglitide analog or anti-diabetic drug. It's used in the type 2 diabetes mellitus. So in terms of absorption, it's rapidly absorbed, undergoes a first-pass metabolism, and bioavailability is 60%. This drug is metabolized by CYP3A4 and CYP2C28. These are the major metabolic enzymes. The key input parameters for this drug is listed here. The parameters which I discussed earlier, log P, pKa, plasma fraction unbound, blood plasma ratio, intrinsic clearance, all of that will be fed into the system. And based on this, and we also assume a permeability limited distribution in the liver. And as I was mentioning, there are two approaches are possible. Top-down approach, that is whenever you have the clinical data, clinical pharmacokinetic data is available. From there, we will go on to predict the parameters. That's top-down approach. When we don't have any of the clinical data or any pharmacokinetic data, we will just build the model from the base from all the in vitro data and then predict the pharmacokinetic. That is called bottom-up approach. Sometimes we combine both this approach. It's called middle-out approach. Okay. In this case, both have been used. Top-down and as well as bottom-up approach has been used. So the drug-drug interactions were simulated using three different inhibitors of the enzyme. Trimethoprim, that is for 2C8, clarithromycin 3A4, and cyclosporine, that is for OATP 1B1. Three different inhibitors are used. And idea is to see when we give this inhibitor separately, what will happen? And when we give all three inhibitors together, what happens? The prediction, that's a kind of prediction goal for this exercise. So initially they will they've started with individual simulations, wherein the effect of each of this drug, each one of this element with this target drug repaglinide was studied. So if you look at this step, we will understand the idea is to establish the relative contribution of three different enzymes to the disposition of repaglinide, and the drug-drug interaction were simulated. These independent inhibitors. Clarithromycin, that's for CYP3A4, that's showed in the green plot here. Trimethoprim, CYP2C8, that's shown in the plot, which is in red color. Cyclosporin, that is specifically the inhibitor of OATP1B1. Compared to the observed data, what's happened with standard doses of standalone drug, with this interaction, what happens? So each of these inhibitors were run separately to identify what is their impact on the AUC of the target drug, which is repaglinate here. And the drug-drug interaction model has predicted the increase in AUC because the inhibitors are there. The clarithromycin predicted with the dose up to 50 mg, it predicted 1.4 fold increase of the AUC of the target drug. Trimethoprim, it showed 1.3 fold increase in the AUC. Cyclosporin showed 2-fold increase in the AUC of the repaglinate. So observed values, these are all like predicted from the PBPK model. Actual observed values from the previous studies showed 1.6, 1.4, or 2.4 fold respectively for these three drugs. They are pretty close. The predicted total, because as of now, we don't know when we combine all these three inhibitors, what will be the total impact? That is not known. But by this PBPK simulation, we incorporated the combined administration of all three inhibitors. The prediction is 5.6 fold of AUC. Are we comfortable with this answer? Yes, pretty much, because we have done the predictions individually and looked at the actual data. The predictions are pretty close. And our assumption is that when this model, all other assumptions worked well, and the last one, the prediction, which is having 5.6 fold increase in AUC, might also be correct. That is our increased confidence level after this combined prediction based drug-drug interaction PBPK model for this drug. So now we focused on the plasma concentrations. That's what when we focused here in this study. Full PBPK models can be used to predict the interactions of this drug in other organs and tissues also. It can predict even in the pancreas, which in this case is the site of action because it's an anti-diabetic drug. The simulated pancreas concentrations can then be used to drive the response because that is where the pancreas, you are starting your pharmacodynamic activity. And that can also be investigated, and we can link this PBPK model into a PD model. So then that model will become PBPK PD model. So you have a PK as well as PD. So that's a kind of potential application is possible with this kind of PBPK models. So when it comes to regulatory implications, the validated models are used by regulators to waive off complex drug-drug interaction studies in the clinics. When we are able to do this kind of model and simulate what will happen in the clinical scenarios, then complex clinical studies are waived off, provided your model is able to answer the questions very well. Also, allow safe initial dosing in children by mathematically simulating the development ontogeny, blood flow, organ size in children. From the adult dose, we are scaling it up to children. That is possible with the use of this PBPK model. And in special group patients like vulnerable patients who are having cirrhotic liver injury or chronic kidney diseases, in those situations also, the PBPK model will be fine-tuned to meet the expectation in the special population, and accordingly, we can talk about the pharmacokinetics in the special populations also. So in all this situation, when we are able to get a good model, we are in a position to get regulatory waiver from conducting such studies in the actual patients. So it's not all that easy all the time. There are issues that need to be addressed before taking it as a very universal system. That is like in vitro systems which are being studied need not always represent or replicate a physiological phenomena. So that in vitro system have to improve so that they can replicate the true in vivo situation. Future developments include combining PBPK model with fully mechanistic PD model, where we understand the mechanism of pharmacodynamic activity. When we link this together, including receptor genotype, we can have a full-fledged PBPK PD models that will be used in a clinical setting to understand the outcomes. Another important area to focus is the predict the disposition of larger molecules, protein-based molecules and large molecules. Their PBPK model have to be established and studied. These are all the necessary further step. And within the industry, the users should continuously train and retain their skill set in the application of this model. And when you look at the PBPK models, they come from very different background from pharmacology, medicine, pharmacy, veterinary science, and mathematics, programming, and statistics. And such a diverse people are working together. There is a very clear-cut need. Communication among this varied group is critical. That's another important area. Communication among all the stakeholders who are working this model is critical. So in future, the drug development is predictive. So we are seeing a lot of examples, a lot of case studies being published year on year. PBPK transcends the empirical observation by using known physiology and mathematical scaling. It predicts exactly why drugs behave the way they do in specific tissues. That's very critical for us to understand and take it further into further PD models. And learn, confirm, refine, loop, continuously stress test the biological assumptions, thereby reducing the late stage clinical attrition and failure. And PBPK is no longer an academic exercise. It is an indispensably regulatory mandated pillar of modern pharmaceutical industry in terms of safety and dosing strategy. So considering all of this, the drug development programs will actively incorporate this kind of mechanistic PBPK models and improve drug development process, leading to, at the end of the day, patient safety outcomes. Yeah, these are the basic references I've taken because it's important to introduce this concept at the basic level with appropriate case study. And that's all I have for this evening. Thanks for listening.
Thank you, Dr. Rajan, for wonderful talk. We'll cover questions at the end of this session. Now, I would like to introduce our next speaker, Dr. Claire Wilson. Claire has over 25 years of experience in drug discovery. At Syngene, she is responsible for driving therapeutic discovery programs from the early stage of target identification and validation through lead discovery and optimization to preclinical evaluation and phase 1 demonstration of safety and proof of mechanism across multiple therapeutic areas and modalities. In this role, Claire supports an extensive portfolio, acting as a program leader and a core team member on several projects. Prior to joining Syngene, Claire held leadership positions at Syneos Health, Galderma, and Evotech. She completed her BSc in biomedical chemistry from Sheffield Hallam University, UK, and PhD in pharmacokinetics from School of Pharmacy at the University of Manchester, UK, where her research was focused on time-dependent inhibition of P450 enzymes. So with this brief background about Claire, I invite Claire to present her talk. Over to you, Claire.
Thank you, Amal, for the nice introduction, and look forward to presenting in this webinar. Today, we're going to talk about the human dose prediction and standard practices in the pharma industry. Several aspects to this presentation. We'll talk about why we need to predict human pharmacokinetics, examine some of the standard practices used in the pharma industry to predict human dose, look at insights into tools and models that can be used in human dose prediction, review the challenges and opportunities, talk about some specialized software used to predict drug exposure, and then I'll talk through some case studies, looking at PBPK as a valuable tool in model-informed drug development and the regulatory decisions involved in PBPK modeling. So first of all, why do we need to understand human pharmacokinetics to aid the selection of optimal dose and dosing regimen for the clinic? We need to understand the human dose to avoid overprediction, which can lead to adverse responses and side effects, and also underprediction, which leads to subtherapeutic doses or no effect in the clinic. And you can see here there's two different graphs looking at the single dose and multiple doses. The desired response is in the green window here. So for a single dose, we want to avoid any adverse responses or side effects, which is shown in the pink on the left-hand side. And also we want to avoid any subtherapeutic effects or doses shown in the yellow. When we move into multiple dosing, we need to be careful because we see potential drug accumulation, which you can see with this once daily dose in the pink here, once daily being dosed at 200 mg, and then 4 times daily dosed at 50 mg. And you can see here that we want to avoid having any adverse effects from drug accumulation. So it's really important for us to understand what is the optimal dose in the clinic to avoid these side effects. So in terms of standard practices using the pharma industry to predict the human dose, the principal goal is to safeguard the well-being of study participants. Hence, drug development is a highly regulated process, and there's multiple regulatory guidelines for entry into human studies. Those guidelines come from the EMA, European Medicines Agency, the FDA, Food and Drug Administration, and the ICH, International Council for Harmonization. And you can see here there's several different guidelines which have been published over the last few years. So in order to look at human dose prediction, quantitative prediction of in vivo PK is essential to choose the right molecules, make the right PK/PD design in preclinical models, and make the right animal to human extrapolations for estimating first in human efficacious and safe dose. So here we see high throughput screening and medicinal chemistry screen the best drugs into chemically similar groups, and we perform adequate in vitro to in vivo extrapolation or IVIVs, and we can predict the in vivo PK from these IVIVs. We can then move into preclinical development and clinical development, where we optimize in vivo PK in preclinical species and relate the Cmax and AUC to the efficacy and safety. And we can use animal preclinical data to human extrapolations and optimize for variability, including drug-drug interactions. So here we see adequate human PK prediction avoids over-prediction, risk of tox, or under-prediction, subtherapeutic doses. We look at some insights into the tools and models which can be used in human dose prediction. 2 generally accepted models are used within the pharma industry: allometric scaling and also physiologically based pharmacokinetic modeling, or PBPK. Allometric scaling really uses the observed in vivo PK parameters from animal models from preclinical species, where we can use simple allometry, simplified allometry, and allometric scaling with correction factors to predict the in vivo PK parameters in human. But we can also use this as a model to understand if we can predict different animal species. For example, you might want to be able to predict the rat PK from a dog and vice versa, to understand if you can get a good prediction across preclinical species before you then move into predicting what might happen in human. With allometry, extrapolation of human dose is based on differences in body weight, and it assumes similar processes in each species. So here we're predicting mainly plasma PK, but of course this is all in animal models. If we then move to the other modeling technique, which is PBPK, this is a mathematical technique that simulates the absorption, distribution, metabolism, and elimination of drugs or chemicals in the body, and it integrates drug-specific data with physiological parameters such as organ volumes and blood flow to predict concentration time profiles. But here we can predict plasma PK, but also tissue PK as well. So it's a really powerful model to enable us to be able to look at tissue and plasma PK. In terms of the PBPK model inputs, we're looking at physiology and compound properties. So generally, we're looking at absorption, distribution, and clearance here. And we can take into account different parameters. So we can look at for example, physiological parameters will be intestinal volume, blood flows, tissue volumes, tissue compositions, enzyme and transporter expression, and also things like the intestinal pH, luminal surface area, and metabolizing enzyme expression when we're looking at the absorption phase. In terms of the compound properties, we're highly interested in lipophilicity, drug charge, plasma protein binding, membrane permeability, tissue partitioning, and of course for absorption we're thinking about the size, the charge, and the formulation of the chemical. So PBPK models combine information on the drug with independent prior knowledge on the physiology and biology at the organism level to achieve a mechanistic representation of the drug in biological systems, allowing the simulation of drug concentration time profiles. Some challenges and opportunities in human dose prediction. So, for example, with allometry, we need to be able to understand all of our PK in vivo data, and also understand if we can integrate the in vitro data generated in discovery to improve the efficacy and speed. We need to understand which animal might be the best predictor of the human situation. Often you've got PK from 3 different preclinical species, and they don't all give the same answers. So you have to try to understand which one is the best predictor, and that can be quite complicated. We also need to understand if we can extrapolate to other compounds and other species and routes of absorption based on the physiology. And what is the effect of dosing routes and formulations? And also, what is the exposure profile in each tissue? And can we even understand the impact of physiology on PK, for example, the age, disease state, or gender? Allometric PK modeling is based on real measured in vivo data, and this may represent a strong advantage, and allometry may capture more accurately the complex formulation effect compared to in vitro or in silico-based PBPK models. When we look at PBPK, this offers an alternative to predict PK ethically, quickly, and potentially more accurately using readily available inputs, for example, in vitro vivo extrapolations and/or in silico data. And we can integrate all of that data generated in drug discovery to extract knowledge and use a mechanistic physiological framework where we take into account intrinsic and extrinsic factors and also the PBPK model components. Generally here, we can look at how to predict, learn from the prediction, and then confirm what might happen in a PK model. So it's a way of being able to do this particular exercise before we even go into a PK study. Once we've done the PK study, we can put that information into the model, learn from it, predict again, and then confirm with another PK study. So essentially, it could allow us to utilize less animals, which talks to the 3Rs opportunity. In terms of the model framework, we can predict blood and tissue PK. So here we're looking at the external dose or exposure, where we can then use that information to give us the internal dose, the tissue dose, and exposure, and also going down to a cellular or subcellular dose level. And here we can look at the cellular-subcellular interaction, try and find an early response and a late response, which takes into account the pharmacodynamics, where we talk about PK/PD, and gives an observable effect. The model is shown on the right-hand side is showing what might happen in terms of the blood flow for each of the different 14 compartments, where we're able to calculate all of the mathematical equations for lung, adipose, etc., to be able to understand what might happen in terms of the whole body situation. And if we look at the graph on the bottom left-hand side, here we're able to see blood or plasma exposure, but also understand what might happen in the liver, the rest of the body, spleen, kidney, for example. So it gives us a really nice framework to be able to understand multiple parametrics in terms of the exposure in different tissues and plasma. How do we apply this PBPK model? So we can look at this in terms of drugs and chemicals. So the pharmaceutical, environmental, and regulatory sciences can use this. And it's now used as a routine approach in several pharma companies for dosing optimization, formulation development, looking at food effects, drug-drug interactions, and bioequivalence studies. In terms of routes of administration, you can do this for looking at the IV routes, translate this to oral, look at IP, subcutaneous, and dermal. And in populations, there's multiple, I've just mentioned a few here. You can look at this in adult populations, translate it into pediatric, geriatric, look at obesity models, different ethnicities, pregnant and neonatal. And of course, there's multiple target organs that we can look at here: liver, skin, kidney, lung, tumors. And future applications, we'll be linking this with PD modeling, and also prediction of drug concentration at the cellular level, looking at nanoformulations, impact of transporters and genetic polymorphisms, the role of CYP enzymes, of course, age, disease states that affect metabolism in PBPK models have been identified. We use some specialized software. Sometimes people have got in-house models. They're able to use different tools such as MATLAB, Berkeley Madonna, and R, which are used for specialized general purpose modeling. There's also some software available from different vendors. Open Systems Pharmacology is one of them. And there's also Satara's SimCip model and Simulations Plus, PBPK Plus models, which is there coming from Gastro Plus. So there's many different ways of being able to predict and use this software. Some case studies which will allow us to look at PBPK as a valuable tool in model-informed drug development. So I've taken some options from the literature and presented some of the papers here, showing some of the images that have come out for looking at novel drugs approved by the US FDA between 2019 and 2023. And here you can see the usage of PBPK models in FDA-approved novel drugs from 2013 to 2023. Here we're seeing that the number of novel drug approvals containing PBPK is increasing, and the percentage of novel drug approvals containing PBPK is also increasing as well from across those years from 2013 to 2023. And if we look at the distribution of PBPK models in applications of novel drugs, we can see that the majority are within DDIs, enzyme transporters, gastric, and also organ impairment models as well, pediatrics, and drug-gene interactions. And the usage of PBPK model platforms in FDA-approved novel drugs from 2019 to 2023, we can see that SimCip was highly used. It's the majority of drug applications. Also GastroPlus, PKSim, and ND, which is non-disclosed, no data for showing what was happening, for predicting and submitting to the FDA. So you can see that PBPK models are rapidly becoming an essential tool to improve the efficiency and quality of novel drug development. And the analysis of current application of PBPK models in novel drugs approved by the FDA in the past 5 years has shown 243 novel drugs from 2019 to 23, 74 applications have used PBPK models. And as we've explained in these pie charts, DDI was the most widely used area of PBPK models for novel drugs, and SimCip was the most popular platform with a usage rate of about 80%. So despite its challenges, PBPK has demonstrated its potential in novel drug development, and submission of PBPK models is becoming routine in the FDA novel drug approval process. This paper talks about a bottom-up and middle-out approaches, where the bottom-up is using a model prediction to generate a PBPK model and then input the observed data, as well as the drug parameters and physiological parameters, and the middle-out approach, where they're using a PBPK model plus the observed data. So the parameters within the model are modified to fit the observed data. This is quite a common way of working because it allows us to have observed data as well as the model to be able to understand what's happening to generate the data that we need to give confidence in a dose prediction, and also the submission to the regulatory authorities. The next paper talks about the evolution and future directions of PBPK modeling in the FDA. It's a regulatory review, and here it's showing data from 2020 to 2024. And it's showing that PBPK has demonstrated a substantial increase in application scope and depth, increasingly adopted by sponsors and submitted to regulatory agents as key evidence. There's multiple draft guidance documents to standardize and promote the appropriate application, and significant advantages in application value in domains such as the DDI and dose selection for special populations. Of course, several limitations still persist: imprecision of parameters, incompleteness of validation, and lack of standardization. But this is something that we can work towards for the future. So in the future, methods such as AI will enhance accuracy of models and unify verification standards to further improve prediction accuracy of PBPK models and provide increased scientific decision-making support for precision medicine and global regulatory strategies. So similar to what we saw with the previous slide here, we're seeing that the use of SimSip as a software platform is around 80%, and also the bar chart is showing we're seeing a huge trend in the percentage of new drug approvals using these PBPK models. Here we're seeing a distribution of therapeutic areas. So in oncology, it's more than 42% of the drugs, followed by the rare diseases. And then we've got CNS, autoimmune cardiology, infectious diseases. You can read the data on the graph yourself, but we're seeing, there's a high variation in the PBPK models that are used for regulatory new drugs. And the application areas, again, a huge amount of work on DDIs, which seems to be the most popular, and then pediatrics, disease, organ impairment models. So just to switch slightly, here now we can look at the physiologically based pharmacokinetic modeling and simulation in regulatory review. This is from an US FDA cyber experience and perspectives. Here we're looking at a comprehensive landscape review of electronically submitted applications to FDA, where PBPK played a role in 26 regulatory submissions and interactions from 2018 and 2024. Applications were from 17 sponsors for 18 products, and 11 of those are intended to treat patients with rare diseases. The PBPK has emerged as a valuable tool in model-informed drug development, and we're looking at opportunities for biological products that are numerous and promising. Optimization of dosing regimens for novel therapies like adeno-associated virus gene therapies, AAV-GTs, and mRNA therapeutics, and valuable in predicting DDIs for new modalities and in estimating drug PK in special populations, for example, hepatic impairment, which is particularly relevant for liver-targeted therapies. So here we can look at the AAV-based GTs as viral vectors for treating genetic diseases by delivering functional genes. And there's currently 8 approved products, and I've listed them here, and they range from different diseases such as hemophilia A and B, and also spinal muscular atrophy, and Duchenne muscular dystrophy. So there is, again, multiple challenges for modeling for cell and gene therapies. Availability of high quality preclinical and clinical data. These are often rare diseases. The complex mechanism of action and high variability in patient responses. Of course, manufacturing variability and potential for immunogenicity, and the acceptance of using these PBPK models as a primary source of evidence for decision making. This is still evolving, particularly for these novel therapies. So this is a really nice paper which shows how we can utilize modeling to help us within this rare disease landscape. And of course, still thinking about there's a collaboration requirement among industry, academia, and regulatory agencies to establish a best practice for model development and application in this regulatory decision making. This is an absolute requirement as we move forward. So the past decade has seen significant advances in basic sciences and regulatory initiatives that facilitate the application of model-informed drug development, including PBPK modeling in drug development and regulatory decision making. So in 2020, the FDA published a white paper on the use of risk-based credibility assessment for PBPK. And in 2021, there was a dedicated workshop on development of best practices in PBPK to support clinical pharmacology regulatory decision making. And in 2024, there was an FDA meeting created to look at the engagement opportunity to discuss the application of model-informed drug development approaches in drug development and regulatory evaluation. This led to the FDA releasing a roadmap that outlines opportunities to reduce animal testing during drug development. In particular, new approach methodologies were described, specifically mentioning PBPK, quantitative systems pharmacology, and ML AI models to leverage existing data to predict safety, immunogenicity, and pharmacokinetics, reducing the need for new animal experiments. And in 2026, the risk-based credibility framework was released, and it's now being incorporated for wider application of model-informed drug development evidence as part of the ICH guideline on general principles for model-informed drug development M15. So we can see there's been a huge amount of advancement in develop the guidelines for regulatory decision making across different governing bodies. So just to finish, there's many advantages of PBPK. It's a mechanistic tool. Tissue PK can be estimated. Virtually all PK complexities can be modeled. There's multiple types of data which can be integrated in silico, in vitro, of course the in vivo as well. Allows direct extrapolation, for example, in vitro and vivo extrapolations, you can look at root to root, low to high dose, animal to human. There's sensitivity analysis built in, so you can look at the variation in different parameters and how that might impact the model. And it's increasingly adopted by sponsors and submitted to regulatory agencies as key evidence. Of course, limitations exist too. They're fairly complex models, and often there's specialized software that's required. Expert user knowledge is needed, and some processes are still not described. Parameters are estimated from in vitro or preclinical in vivo data, which may not accurately reflect the human in vivo conditions, and there's challenges concerning validity, transparency, and lack of international consensus on validation protocols and performance criteria. But of course, saying all this, within the next 5 to 10 years, we'll likely see that these models accelerate the time to market for new treatments and provide more scientific decision-making support for precision medicine and global regulatory strategies. So we look forward to seeing how this develops in the next 5 to 10 years. Thank you very much.
Thank you, Claire, for insightful talk. And now with this, we'll open a question and answer session. So first question is for you, Dr. Rajan. It is from Dr. P. Anshuman Abik.
Okay.
He's asking how computer-aided design of phytochemicals can be combined with PBPK modeling in cost-effective way in resource-constrained settings, especially in tertiary care teaching hospitals.
Okay. Of course, too many questions targeted into one question. Yeah, phytochemical constituents. When we say the one question we have to understand is sometimes the usual research here is like they take an extract of a particular, imagine a herbal portion. Sometimes it contains multiple components. Unless if we are able to purify a specific component, it acts like a chemical, any other chemical agent. Then you know the structure of the compound, then physicochemical properties can be derived, and then that can be used in a PBPK model, provided we know the exact chemical structure of the compound, phytocompound, which we are talking. Once you have that, the same way it will work. So until the level, with all the available information, we can get into the PBPK model. But what happens when you have a new compound? You may have to do that experiment, lab-based experiments to get the physicochemical properties data, how much you can predict from certain softwares or tools, but still we need experiments to verify these values. Once you have that kind of information, then you can use that as a input into this model, and all the available physiological information, everything available, and this softwares will predict how the drug will distribute and then how that will get into the system. It's quite possible, provided we know the structure of that compound very well, the phytoconstituent which you're talking. It works like any other agent. It can work. Should not be a problem.
Yeah, thank you. So other question is from Dr. Karthik Armugam for you, Dr. Rajan.
Yeah.
How did you verify the model performance for repaglinide? What are the model evaluation parameters and how did it compare against observed data? Any exposure different between healthy and patient population?
Okay, fine. Here, this case scenario, it's available as I've given the reference also. It's available as a case study. So what they have done when they started this one, they studied the repaglinide, the effect of this drug inhibitors independently. They have studied separately, and
Then they combined what is the relative contribution of each of this inhibitor to the total level of contribution. Like that, we can they verified. So that's how they are saying the independent simulations and combined simulations. Then when it comes to model qualification, we have a criteria. Like for example, you have observed data is available, clinical data or animal data is available. When we scale up from bottom up approach, when we come with a prediction, there is some kind of 50% rule we'll have. For example, whatever you predict should be 50%. At the max, it could be 1:2 ratio. They will allow until 50% to 100% variability is possible. Within that window, we are able to predict from the bottom-up approach. It is considered as some kind of acceptable number. Okay. Then with that, if you're not happy, you can always fine-tune the model parameters to reach the level of accuracy. That kind of model evaluation mechanism is there within the PBPK model approach. Okay, that's possible.
Yeah. Thank you. Next question is for you, Claire. In your experience, what kind of molecules allometry works well and what doesn't work? Any examples can you share?
Yeah, so that's an interesting question, because what I discussed in my talk was really about in vivo situations can be really useful for allometric scaling. But of course, if the rat predicts the dog and the dog predicts the rat, for example, then you've got a good chance that the in vivo situation in the human might be similar to those species. But sometimes when you integrate a third species, for example, non-human primate, you might actually get that the data doesn't all match up together. Therefore, it's very challenging to understand which preclinical species would match the best in terms of PK, especially if you're talking about small molecules. So in this sense, it's always good to do the allometric approach if you've got 2 to 3 preclinical species, but also use the PBPK approach as well to see which method gives you the data, which I guess if the both methods match up, then you've got confidence in the both of the models. So it's kind of a touch and go situation. There is no one animal predicting best. It's a combination, and it's a combination of models as well. Hope that helps.
Yeah, thanks, Claire. So next question is also for you from Govind More. He is asking, could you further discuss how PBPK modeling can be applied in the development of 505(b)2 drug products, particularly in scenarios where the molecule exhibits higher plasma concentration than the lowest effective dose at low dose level and possesses a long elimination half-life?
It sounds like from the question that the new drug may have some issues in terms of safety or efficacy. And really, we can use PBPK to apply the 505(b)2 development pathway. It's a bridge, basically, isn't it, between a new product and a previously approved reference drug. So, of course, we can apply that as an opportunity, but we have to justify any PK differences. And the PK differences here, it sounds like it actually has an impact on the safety or efficacy, given the question, if I've understood the question correctly. So I would be quite wary in just applying a model to say, well, this is what we've got. It sounds like it's higher plasma concentration than the lowest effective dose, and also a long elimination half-life. I'd be quite careful. Maybe you could do some further studies in vivo to understand exactly what's happening and work out if you can actually apply some sort of model to give you some confidence going into the clinic, for example. But the regulators would be advising here on what to do and whether they'd accept that model as an opportunity for using the 505(b)2 pathway.
Yeah, thank you. So next question is for Dr. Rajan. Can we possibly account or model for alterations to blood flow into any organ due to any disease states?
Yes, it's possible. That's what, right? In case I explained in special scenarios like renal failure or hepatic failure, when the organ dysfunction happens, if we specifically know that information, for example, hepatic failure, a particular of cirrhosis, we know what will happen to the blood flow in those scenarios. That, and anyway, organ blood flow is an inherent part of that organ related, system related, that can be modified. We go into the system, we can perturb the system or change the system according to the input, then the model will account. And as well as hepatic and renal failure scenarios are also already inbuilt in many of the already available tools. So that way, this can be accounted for. That's how this is exactly PBPK model is supposed to do. When we have organ dysfunction, that will be specifically accounted in your input system, that will account and take care of that particular issue.
Yeah, next question is also for you from Priyambada.
Yeah.
Question is, can I convert the HIM, human intestinal microsome data, into KM and Vmax for CYP3A4 as the abundance is high in intestinal microsomes?
Yeah. Yes, and I would I would be happily post this question to Claire also with her practical experience. He can also add some point here. Definitely that when we have this CYP3A4 specific metabolism pathway is there in our PBPK model, in that organ system, we are actually can give that information. For example, CYP3A5, CYP3A4, whatever the CYP enzymes, we have an option to scale or give the information about the how much this enzymes are available. And that information are specific to even ethnic groups also. That can be fed into the initial, when we model setup only, we can set up based on the Caucasian population or a specific population. When that information is available, the model will scale that information into that enzyme specific, based on that Km and Vmax is calculated. And that backend calculations happen in the model, in these models.
Maybe we could also add here that some models for, especially related to CYP enzymes, have also included a part of a parameter called K degradation, and it's looking at the degradation rate of the enzyme itself and the rate of de novo synthesis, which for, I'm not sure about the intestinal CYP3A4, but certainly for liver CYP3A4, and I imagine it's similar, it can be up to 24 to 48 hours. So there is an opportunity here to add that in as the model as well, to understand if it's, if for example a drug is being administered, it could also act as an opportunity to inhibit that drug. That's not part of this question here, but I just thought I'd add it in as an extra example for the modeling.
Yeah, thank you, Claire. So one last question in interest of time. Maybe Dr. Rajan or Claire, either of you can answer. It's from Vishal Pathade. How do we optimize the parameters for complex moieties such as degraders using PBPK modeling approach? Which software would be helpful for academic, as a commercial softwares are costly and it is difficult to learn and get hands-on for new learners?
It's a really good question, and it's something that I haven't actually had experience with in terms of looking at the degraders. But I should imagine that since there's some cheaper software options available, especially in the academic environment, such as the Berkeley Madonna or maybe R, that could be somewhere to start with those modeling techniques and obviously publications around those. I understand that the commercial software is really expensive and it's really challenging to get hold of that and to start learning using that. So maybe the other software platforms I mentioned might be of use if you've got them available in your academic environment.
I think the question is pertaining to the PROTAC and those kind of molecules.
Yeah.
I think, yeah. I think in terms of publication wise also, I understand very few publications are there in this domain. There is a need to work on this, I understand. In terms of, I think, commercial tools, at least something which is available, PKSIM is one tool, which is a little bit easy for the beginners to start. And Berkeley Madonna, as you said, Dr. Claire was mentioning, yes, Berkeley Madonna and R-based platforms are also now increasingly being, shown up by the other groups. there again you need to tweak in the model, write your own codes, all those things. That makes little harder for a beginner in the case. So we have to explore possibly how PKSIM, which is almost like your GastroPlus tool, is able to work in this scenario. So I think there's a need for certain research in this domain, and there are not many papers are available as of now.
Yeah, that's true. Thank you, Dr. Claire and Dr. Rajan. In interest of time, we'll respond to remaining questions via email. So I would like to take this opportunity to thank Dr. Rajan and Dr. Claire for taking time out of their busy schedule to deliver today's talk. I also extend my gratitude to Dr. Priyaranjan Patnaik, Dr. Anwar Murtuza, Dr. Jayesh Iyer, and the senior management of Syngene for providing this valuable platform to share knowledge. My sincere thanks to Syngene marketing team for their support in organizing this webinar. Finally, last but not least, I thank you all participants for joining us. We'll upload the webinar recordings on the Syngene website and on our LinkedIn webpage. Thank you once again for attending the session. We look forward to seeing you soon with a new topic. Until then, take care. Bye-bye.